Alright, so a little bit forward, today we're going to be looking at genetics. It's our first class in genetics. It's going to be fun. I believe it's going to be awesome. Genetics is a long unit, suit yourself. So we're going to look at what is DNA first. So today is pretty much a review, but still we're gonna learn new stuff. So inside of a cell, you'll find this nucleus, and inside of the nucleolus, you will find these chromosomes. And that's what dictates, well, how we make proteins, essentially. So DNA only codes how you make proteins, and that's it. You thought DNA was all of that fancy life code? Yes, it is. However, it's more of just talking about, hey, how do you function? So DNA is, what we call chromosomes, basically. So DNA is in chromosomes, and chromosomes can be further broken down into chromatins. And these chromatins, they could be broken down into DNA structures, or double helix structures. That's what we call it. So when we look at these double helix structures, we will see, when we look at it through a microscope, we will see that these genes, it contains these nucleotides. These nucleotides are, well, we only learned 4 in grade 11, but there's another one. So we have the guanine, cytosine, adenine, and thymine. These will be arranged together in the form that A will bond with T and C will be with G. And you can see it, ATCG. ATCG is never going to be different. It's always going to be ATCG. So the gene tells you what this cell will do. It will control the structure and control what will happen. So basically the what, why, and how, and future. Humans, we have 46 chromosomes, obviously, and to say 23 haploids, basically. So we're going to look at the cell cycle. The cell cycle you learn in grade, probably grade 9, hopefully grade 9, that there's 3 main cycles. Well, there's 3 main parts, obviously. There's growth, repair and replace, and reproduction. Growth, DNA replicates, and DNA actually dividing. So we can see we have the interface. That is the start of everything. In the interface, we will grow and grow and grow and prepare to divide. And finally, we have the mitosis. So you can say that all of these, this, and this, they're all part of interface. You have the growth, you have the replicate, and then you have preparation for cell division. All of these are interface. And then you have only 45% of it is mitosis. Or to say, sorry, not 45, only 25% of it, or even less, is mitosis. And in mitosis, it's further divided into 5 groups. Well, not including cytokinesis, then it's 4 groups. You have the prophase, metaphase, anaphase, telophase, and finally you end on cytokinesis to check that you are actually a working cell. And the end of this cell cycle that you learned in grade 9 and grade 10, you will produce 2 exactly completely perfect identical daughter cells. They will not have any different, they are not able to be differentiated from each other. They're completely identical. They're, most of the time, they're extremely perfect. And you can't tell which was which. So let's look at the interface. Interface, it's basically the periods of growth in the life of a cell. So it keeps on growing, it grows, it replicates, and it has 2 growth stages and a DNA replication stage. So you have growth first, and then you have DNA replicates, and then it grows again, and prepare for cell division. So we look at how replication is actually done. We have this chromosome that's consisting of 1 chromatid. It duplicates. It becomes a full duplicated chromosome, and it has this 1 dot in the middle that connects both of them. And that's what we call sister chromatids. And then mitosis. This is the process which the duplicated content of the cell's nucleus will be divided into 2 equal parts. Well, you might come up with a question. Can the cell just divide down in the middle? That's a question. That's no. You see, here we have a full video. PMAT tells you what stages of mitosis: prophase, metaphase, anaphase, telophase. So let's look at this animation right here and observe it. Prophase, metaphase, anaphase, telophase. So it quickly aligns themselves, that's metaphase. It grabs on, and that was prophase, and now it separates. Okay, that was prophase. This is metaphase. It lines up. Anaphase, it starts pulling. Telophase are done. Alright, so we look at prophase first. So pro means before in Latin, and chromosome, chromatin in here, it coils and condenses into chromosomes. Nuclear membrane starts to break down, and nucleolus starts to disappear. We can't find them. Centrioles head to the opposite side of the cell. You see, these are centrioles, are heading to the opposite side. And spindle fibers will form between these centrioles and grab onto each other, and all of these, they will eventually attach to them, but that's in the metaphase. So metaphase, it's mid in Latin. Centrioles both reach the end of the cell, the double ends of the cell, and chromosomes, they will line up in the middle, and the spindle fibers from the centrioles will attach the centromere. So remember, you have these things connecting them together. So the spindle fibers will attach to the centromeres that's connecting them together, so that the chromosomes obviously stay there. So finally, we have anaphase, where we start pulling. Ana means back in Latin. So spindle fibers, they will start pulling these sister chromatids apart. And so when they pull apart, they're going to be newly split, and they will move to the edge opposite of the cell, which you can see here in telophase. Telo is end in Latin, and this is when the spindle fibers is going to start to disappear, because your mitosis is over. It's done. The nuclear membrane will reform and come back, and it will form around each set of chromosomes. Nucleolus reappears, and chromosome becomes less coiled, so it goes back to chromatins. Now, that was mitosis. That was quick because it was a review. So we're going to look at the idea of meiosis. So, meiosis, it's basically mitosis. However, meiosis only takes place in a gametic cell. So you gotta know, mitosis is mitosis, alright? Hear my pronunciations and hear my words because it's gonna confuse you. Mitosis takes place in somatic cells. And somatic cells are everything that's not a reproductive cell. You just gotta know that. You could be a skin cell, a muscle cell, or whatever. As long as you're not a reproductive cell, you're not a sperm or an egg, then you will do mitosis. However, if you are a sex cell or reproductive cell, you are always going to be doing meiosis. So meiosis, we have prophase 1. The chromosomes become visible under a microscope as they condense. They will all of them, they will consist of their sister chromatids. So remember, one from dad, one from mom. This process is called synapsis, and it will form tetrads. So when you have 4 of these chromatids, it's called a tetrad. So look at this. This is not a perfect tetrad. It's not like they just lay there. No. Some section of this DNA will be crossing over, exchanged, so that we actually have genetic diversity. So these homologous chromosomes at points called chiasmata or chiasmata or kismata, they will lead to genetic recombination. So that is essentially why that when your mom and your dad sexually reproduces, you are not going to be perfectly like your dad or perfectly like your mom. You're going to have this crossover, and then essentially you're going to be like partially like your mom or partially like your dad. So we're going to go into metaphase 1. And keep in mind, there's a number here. So this is basically the same thing as mitosis. However, now you have all of these tetrads sitting here. So the paired tetrads, they will align themselves along the cell's equator. So it's going to be in the middle. It's going to align themselves onto the spindle fibers in the very center middle. This is kind of like a 3D image, so you gotta look at it a bit 3D. You gotta look at it a bit abstract. So you see, these are the spindle fibers. They will pull them. They will pull. Remember, they also attach themselves to the centromeres. Now they're gonna pull. So right here you have anaphase. They attach themselves to centromeres. Now they're pulling. Now, when they are pulled, they become sister chromatids. So now there's only 2. Remember, the sister chromatids from mitosis. So the spindle fibers will shorten. They will pull these apart and to the opposite side of the cell. So it's basically the same thing as anaphase for mitosis, except you're having these sister chromatids. Each pull will receive a random assortment of maternal and paternal chromosomes. it will still contribute to genetic diversity. So remember, when you look at these, there's always going to be blue, red, or red, blue. There's always going to be some sort of modification to them. They're not going to be exactly perfect. Because if they're exactly perfect, we don't have genetic diversity and we die. Now, telophase. See here? Look, telophase, new cells are formed. So homologous chromosomes reach the complete pole opposite of the cell. The new nuclear envelope will form around the set of chromosomes, and the cytoplasm will divide. You had a lot of cytoplasm here. Now they fill in only these two. They will result in 2 haploid daughter cells, each with half of the original chromosome number. So remember, haploid and diploid. Haploid has 1 set of chromosome, diploid has 2 set of chromosome. That's the difference. That's it. You just gotta remember, that's so simple. The cell will now enter a brief interphase like period before preceding meiosis 2. So it will grow a little bit and then will prepare to divide. Now, what is mitosis 2? What is meiosis 2? See, now the numbers are adding up. This is prophase 2 and this was prophase 1. So prophase 2, you have, remember, you produced 2 daughter cells. Okay, so keep the numbers in mind. You have 2 daughter cells here. Each of them has a sister chromatid. Sister chromatids, remember 2? And now it's gonna go through all of these processes again, and they will pull each other apart like this. So the nuclear envelope is gonna disappear. The spindle fiber is gonna form. They're gonna attach themselves to the spindle fibers. The spindle fiber is gonna pull them, and now they're apart. And now look at what happens. You produced 4 genetically unique, and when I say unique, you gotta understand, you produced genetically unique cells. You produced 4 genetically unique cells. 2 divided into 4, and each of them only has 1 strand. So meiosis is basically mitosis, but instead of just purely creating daughter cells, you're creating genetically unique cells. And on top of that, you're creating 4 instead of 2. So mitosis only creates 2, but meiosis creates 4. That's the major difference. So now I want to talk about something more, a little specific. So the big thing that I want you to understand from meiosis is that this is going to be crucial. This is going to be on your test. This is very important. Is that it produces. Okay, so meiosis. Let's do a compare and contrast. So meiosis only works in sex cells, gametic cells, reproductive cells, while mitosis only works in somatic cells like skin cells, muscle cells that do not have sex organs and cannot pass that gene on to the next generation. The big major difference is meiosis creates 4 genetically unique haploid cells, and mitosis creates 2 genetically completely identical diploid daughter cells. Perfectly perfect. There will rarely be any mistakes. So essentially that's all we have today. Remember, it's a review. We're going to look at more interesting stuff next class.